Welcome back, students. So I Raminder Kaur. Here I will start my fourth video lecture. So the topic from chapter number 9 is plant breeding for improved food quality. As we have studied animal husbandry and different procedures, that how the schematic steps are being performed in plant breeding. What is the objective behind it? We know that the population is increasing day by day and many people are hungry. So, to serve them, they are eating the food, but that is not a balanced diet. How can we enrich their diet with good nutrients that can be carbohydrates, proteins, fats, vitamins, and minerals? So, scientists take out a technique known as biofortification. So, on your page, here is biofortification. So, if I start, let's first of all discuss what are the aims behind plant breeding. So if I focus here, you can see number first, high yield. We want to improve the quantity. Number second, better quality of produce. The produce can be any fruit, it can be vegetables, or it can be crops such as cereals, pulses, etc. Number third is increased tolerance to environmental stress. We have studied lot many stresses in the previous videos. It can be drought, it can be floods, salinity, high exposure to light, high exposure to temperature. It can be high, it can be low. So we want those kind of crops which can grow under any critical extreme situations. Number fourth is resistance to pathogens. Pathogens are those microorganisms which cause infection either in plants or animals. In case of plants, there are several pests, several insects, and microorganisms such as bacteria, fungus, protozoans, nematodes, which cause reduction in the yield as well as the quality of the crops. So, we have studied several varieties and hybrids of crops by which we can make them resistant, so that we should have to spray very less amount of fertilizers as well as insecticides and pesticides. So last is also same, increased tolerance to insect pest. So this topic biofortification talks about how the hidden hunger could be stopped. Hidden hunger means those people who are eating food but that is not nutrient rich. I'm repeating, hidden hunger means that diet which lack adequate nutrients. That can be micronutrient, that can be proteins or vitamins, because they don't have enough money to buy or afford these kind of nutrients in their diets. So what can be done, that we can increase the quality of protein, oils, vitamins in normal growing crops. So particularly iron, vitamin A, iodine, and zinc. They are deficient in diet and they can lead to reduction in lifespan, more prone to diseases. That means immunity gets slowed down and several kind of mental abilities can arise. It can be dementia, it can be depression. So body gets more prone, there's a more risk of infections in those people who are not taking balanced diet. So let's start with our topic, very interesting, biofortification. So scientists or breeders, we can say, they have incorporated higher level of vitamins, minerals, proteins, and healthier fats. Note down everyone, healthier fats, there are two kinds of fats, saturated and unsaturated. You are studying this in chemistry also, that those fats which are healthier, which can easily digested for in our body, that is called as healthy fat. Now, what are the objectives behind biofortification? Here you can see it, protein content and quality. So from the starting we are talking only about quantity and quality. So number first, it is of protein. Number second, oil. Number third, vitamins. It can be A, B, C, D, E and K. Number fourth is micronutrient. Now the term micronutrient means those nutrients which are required in trace amounts, minute amounts, and the mineral content. So here the picture, if we focus on this, latest biofortified crops in the market are number one, cassava, rich in provitamin A. Number two, beans. We will also discuss the type of beans further. Iron rich beans. Number third, maize, rich in provitamin A. Pearl millet, rich in iron. Next, sweet potato, rich in provitamin A. Rice, rich in zinc. Wheat, rich in zinc. You can also see that there are name of the countries written, means from where these biofortified crops are being made. Now, let's come to this paragraph. Here they have talked about that in 2000 maize hybrids that had twice the amount of amino acids. If we talk about that, these proteins, the proteins are only made up of amino acids. Amino acids such as lysine and tryptophan, they are rich in maize. All the corn crops, they are rich in this lysine and tryptophan compared to the existing one. So how in 2000 they are increased? With biofortification. If we come to brief of biofortification, how it is done? The genes of desirable quality, they are taken out and they are incorporated and hybridization is performed. If you remember all the steps of plant breeding, all those are same. Number first was collection of variability. Germplasm collection was there. Then number second, we will identify and we will select the parents. Only those parents with the desirable qualities. Number third was hybridization. Hybridization means mating, crossing, recombination. Number fourth step is testing. Why the testing is done? So that we can check all the desired contents are there or not. And last step is when the test is being done, then we can release it, we can commercialize it, and everybody can buy it. So let's come back. We are here talking about biofortified crops. Number first, we have studied maize hybrids that are made rich in two amino acids, lysine and tryptophan. You have to learn the names of these amino acids. Number two is wheat variety. The name is Atlas 66, that is made rich with high protein content. Then iron fortified rice. This is the number third. Put number 3 on this. Iron fortified rice. We have also studied earlier in zinc as well as iron. So they are made rich in rice. So that is 5 times increased in the earlier consumed varieties. Here also you can see, if I zoom this, you can see so many crops are there which are being biofortified. Next, Indian Agricultural Research Institute, New Delhi. It is also known as IARI. So you must know the abbreviation forms also. It released number of crops that are biofortified. Number first, vitamin A enriched carrots. We all know vitamin A is important for eyes. So carrots, they have made more enriched in vitamin A. Spinach, you all know, that is also with vitamin A. Pumpkin. Then comes bitter gourd. They have incorporated with vitamin C. Bathua. Bathua is mostly used in saag preparation. You can see here I took a picture of bathua. So this is bathua. Chenopodium is the name. Chenopodium. Chenopodium. Yes. So bathua, chenopodium. So chenopodium is a scientific name of bathua, right? So in this also they have enriched vitamin C. Then is mustard, then tomato. Then they have talked that biofortified spinach and bathua rich in iron and calcium has been made. And last is most of the beans. I'm repeating, most of the beans such as broad beans. So here you can see a picture. Flat beans I have shown, lub beans I have shown, broad beans I have shown, and garden peas. All of you are familiar with garden peas, the peas which we are eating. So they are made protein rich. So this was the topic of biofortification. Now the next topic which we come to is single cell protein. As the topic clear, single cell means when the single cell is taken and that protein can be taken up as a diet, it is much nutritious as compared to any other food. Single cell protein. Conventional agricultural production of cereals, pulses, vegetables, fruits may not be able to meet the demand of food. Why? Because population is increasing. That's why we have to shift from grains. All the grains which we are eating, either you are eating a dal, pulses, that is a grain, cereals you are eating, right? So to meet diets also create more demand for cereal. Listen very carefully, this is important and critical question, right? If we say we intake less grains and we turn our diet from vegetarian to non-vegetarian, we can reduce the consumption of the plants such as produces. But this is not true. Why it is false? Because when we talk about poultry farming, we need broilers for meat. What we will feed them? We need a meat, but we have to feed them for grains. So it takes 3 to 10 kg of grain to produce 1 kg of meat by animal farming. Instead of animal farming, you can also write poultry farm. So can you explain this statement in the light of your knowledge of food chains? So answer is because those chicken, those hen from which we are taking meat, they are also depending upon grains. That's why we can't go far from this condition that food chain is there. Either we eat vegetarian diet or non-vegetarian diet. Ultimately, we are dependent upon the plants, that is grains. So more than 25% of population is suffering from hunger and malnutrition. So the solution could be single cell protein. So what is it done inside this? You can see here microorganisms are used for the production of single cell protein. Number first, fungus. Number two, yeast. Number third, algae. Algae is very common because algae grows by its own. Spirulina is a very important microbe that grows by its own, and it only requires very cheap raw material for its growth. And last is bacteria. So if the question arises in your mind here, can we eat microorganism? Answer is yes. Not all, but those which are selected ones which are acting as single cell protein. So let's read in brief. Microbes are grown on an industrial scale. Why industrial scale? What is the meaning of industrial scale? Industrial scale means at a large scale, and it is used as nutrient rich food. Example is spirulina. What are its advantages? Why we are talking about all these different technologies? Number first, they are rich in proteins and low in fats. You all know fats digestion is very difficult by our body. So they are rich in proteins. Number second, they can be easily grown, as I already talked about. They can grow on wastewater. Now the question may comes in your mind that if we grow these kind of microbes on wastewater, do the toxins enter through the xylem? Answer is no. The roots can select which minerals it has to absorb and which not. All the toxins will remain in wastewater and all the useful substances are taken, and then the growth is done. Number second is animal manure. So all are familiar with animal manure, the excreta, the dead and decaying matter of the animal waste. Last is molasses. Molasses is sugar cane raw material, the leftover part of the sugar cane from which sugar cane juice has been extracted. All have seen when we are drinking those sugarcane juice in summer season, so the leftovers are called as molasses because they also are rich in diet. Microorganisms can grow with that also. Next is the use of wastewater reduces pollution. So water pollution will be reduced and they act as a supply of fertilizers as well as pesticides. So this was the main of single cell protein. Now let's see. It has been calculated that a 250 kg cow produces 200 gram of protein per day. Means according to per unit it is less. but in the same period we can get more proteins from 250 gram of microorganism such as Methylophilus methylotrophus. Methylophilus is generic name, Methylotrophus is species name. Why? Because its biomass production is more. Have you seen the pond which is fully covered by algae on the top? So, if we calculate the biomass production of these microorganisms, that is always more. So, it can produce 25 tons of protein. So, it is above from our imagination also. So, they have talked about the mushrooms which we eat and the large scale mushroom culture that is growing day by day, and we are accepting it as a food. It is a fungus, but it is not injurious to our health. So now we come to the last topic of the chapter, that is tissue culture. If you see this picture, plants are grown in the test tube. In the laboratory, if we are growing them, what can be the advantages? Advantages can be we can maintain the environmental conditions. We can give them proper nutrient medium. Then we can grow them at a large scale in a less time. You can see the white color solution that is called as nutrient medium. This is not only water, because water may have few dissolved minerals, but for the growth of the plant it requires something that should be extra, such as soil. So here soil is not added in it. If you remember in class 11th mineral nutrition chapter, we have studied hydroponics. It is similar to that, but few variations are there. Hydroponics means growing plants without soil, only with water. But water should have some nutrient medium. It is a mixture of different plant hormones. If you remember, we did auxins, gibberellins, cytokinins, abscisic acid, and ethylene hormone. So, this nutrient medium is rich with auxin, cytokinin, and gibberellin, right? But how it is grown? So, let's read it. Tissue culture is a technique that is started by scientists in 1950s, and the whole plant could regenerate from explant. Now what is explant? If you see this picture, they have shown explant is a small part of any food or any plant. Explant, a small part of a plant that can be put in a nutrient media. It can grow. Growth means divide, because these are the few cells only, right? So when those cells grow, that is called as callus. When that callus grows, called as embryoid. Embryoid turn into embryo, and embryo turns into plantlet. If you can wonder that how can a small test tube gives rise to such a carrot they have shown here. So these are not the small test tubes, they are bigger one. So here only for reference in the picture it is shown, right? So if we talk about now schematically, number first, it is a technique of regeneration of whole plant from any part. Now here part can be a meristem also, part can be a leaf, it can be stem, it can be root, it can be a bud also. It should be grown in a suitable culture medium. So I'm repeating, suitable culture medium means nutrient medium that should be containing some of the hormones. Plant hormones are also known as phytohormones. So they should contain adequate amount of those minerals, vitamins and hormones. In vitro, if you remember, I have already told you in vitro means artificially, because here manually we are doing it. It is not a natural technique. Aseptic condition. Aseptic conditions means under hygienic conditions. What are the advantages? As we have already studied, number of plants can be grown in a short period of time. Means quantity can be improved. Second is healthy, disease-free plant can be grown by meristem culture. This is also important part of this tissue culture technique. Let's see. A plant is suffering from a disease. Can we grow a new plant from a suffering plant? Answer is yes. Now the question comes, how can we grow? It is suffering from a disease. Let's say a fungus has attacked any plant. If we take a meristem, you remember meristematic tissue, apical meristem, intercalary meristem, and lateral meristem. Meristem is that tissue which is continuously growing, but it is not a differentiated tissue. It cannot perform any function, it is only dividing. So, if we take a meristem of any infected plant, also, we can grow a disease free plant. Can we call it as a healthy plant? Answer is yes. So tissue culture technique is very important. If a plant is infected, we can grow a new plant from it, from its meristem. Last part is somatic hybridization. So we will study it. Do not confuse. Let's talk about now few terms which are given here. I hope you are clear with explants. Any part of a plant, it could be stem, it can be leaf, bud, roots, which can be grown in a test tube under sterile condition. I'm repeating, sterile condition means hygienic condition. Nowadays, as virus has spread everywhere, so each and everything is sanitized. That is also called as sterile in special nutrient media. Now, next term is totipotency, the capacity. Any part can grow into a whole plant. That capacity is called as totipotency. Now, what has been added in the nutrient medium? Number one, sucrose. Sucrose is what? Sugar. Sucrose is a rich in nutrients, right? What else has been added? Some inorganic salts, vitamins, amino acids, growth regulators. What are growth regulators? Phytohormones or plant hormones. Auxins, cytokinins, etc. etc. means it can be gibberellins, ethylene, abscisic acid, etc. The method of producing thousands of plants through tissue culture is called micropropagation. Why called micro? Because we have used a small part of the plant to grow the whole plants. Do you think these plants will differ from their parent plants? Answer is no. If you remember the asexual and sexual reproduction differences, this technique is coming under sexual reproduction or asexual reproduction? Answer is asexual reproduction. So soma clones will be there. Soma clones means clones. Clones means which are duplicates of the parents. There are no variations at all. Which kind of food plants are coming under it, which are being made by tissue culture? Tomato, banana, apple. So they are commercially produced. Now we come to the second part, that is meristem. We have already talked about we can make virus free or disease free plants from any infected plant if we use meristem. Here it is termed apical and axillary. So I have already talked apical meristem and lateral meristem. So we can just take the meristem, we can grow it in the test tube with suitable nutrient mediums, and a virus free plants can come. So this technique has been cultured in banana, sugarcane and potato. Last part of tissue culture technique is somatic hybridization. If we talk about here, can you see in this picture there are two cells which are taken. What we will do, we will take a protoplast. If I here revise, cell is covered by first of all cell wall, then plasma membrane, then what is present inside everything is called protoplast. If I talk about in the center, nucleus is there surrounded by cytoplasm, but protoplast is cytoplasm plus nucleus, means everything inside the plasma membrane is called as protoplast. If we made the fusion of protoplast of two different cells, it can lead to hybrid somatics. This somatic hybrid can be grown into a new plant. Fine, let's brief it out. In this case, somatic hybridization, protoplast of two somatic cells. We all know there are two kind of cells in the body, somatic cells and germ cells. Yes, somatic cells are those cells which are never participating in reproduction. They have their own rules, right? So if any two somatic cells, it can be leaf somatic cells, it can be stem somatic cells, if we fuse the protoplast of either same varieties or different species also, then it can lead to somatic hybrids. Step is, first of all, isolation of protoplast. How can we isolate? We have to break the cell wall. Cell wall can be broken by few enzyme reaction and protoplast can be taken out. Number second is fusion of protoplast. It is again done in laboratory. All the three techniques are done in laboratory only. Number third is growing those fused protoplasts so that somatic hybrid can be made. Now one example is written, pomato. Have you heard of it? If potato protoplast and tomato protoplast is fused, we can get pomato. But there is no further intercorporation is there that we cannot commercially use it. This technique is there, but unfortunately, it has not resulted desirably. It has not given us desirable results because if we say ethically, maybe it is not true that we can take two different species and we can result into something else. So here with this note I am completing this chapter. So today we have studied biofortification, then tissue culture, then meristem, then last topic we have studied is somatic hybridization. So after this you have the assignment to complete these NCERT questions in the notebook. So 12 questions are there. Many of these questions I have already given you when we have talked about the very first or second video lecture. PDF document of important questions is attached with this last video. You have to complete all those question answers in the fair notebook. Note down, there are 1 mark, 2 mark and 3 marks questions. Answers are also attached with them. Complete your notebooks and we will meet with the next chapter. Thank you.